So thank you everyone for joining today. Today we have 10x Genomics will be presenting about their most recent updates and kind of all of their portfolio that we can use for different single cell applications. But we'll be starting with Pat Soteropoulos from our Genomics Center, who'll be talking a little bit about what they offer, particularly in connection to the 10x offering. So Pat, if you want to start taking it away.
Sure. I'm the director of the Genomics Center. I'm Pat Soteropoulos, and the Genomics Center and the Molecular and Genomics Informatics Core are really happy to be presenting this technology series and to be hosting 10x Genomics today. For those of you who don't know about the Genomics Center, we've been here for a really long time with various names over the years. I've been here for 22 years running genomics at New Jersey Medical School. And we have a bunch of labs providing various genomic services, including research genomics, clinical genomics, which includes a clinical diagnostic service, a support services lab called the Molecular Resource Facility, and also the new emerging pathogens clinical lab that was founded with the start of the pandemic, which provides COVID testing, sequencing, and also point-of-care testing. All of our equipment is available for use for both research and clinical diagnostic applications. We provide fee-for-service, research collaborations, experimental design consultation, paper and grant writing support, and data analysis support. In particular, we're collaborating very closely with MAGIC, the Molecular and Genomics Informatics Core, and ALEX to provide full support for all the data analysis for the sequencing data generated. We have a lot of instrumentation. We have a full suite of Illumina sequencers, including MiniSeq, MiSeq, NextSeq, and NovaSeq. We have recently acquired, actually not that recently, it's about a year now, the Oxford Nanopore sequencer. And we also provide Affymetrix and Agilent microarray services. We can support pretty much any protocol that any of these instruments has. We do RNA sequencing, DNA sequencing. We can do whole genomes, whole exomes, targeted panels, a lot of single cell sequencing, and 10x is going to talk all about that today, as well as spatial transcriptomics through Visium protocol. And we also do array CGH, which is particularly useful in the clinical diagnostic service. We have a really strong group of researchers who've been here probably on average 15 years. And we're all here to provide services for Rutgers researchers, but also any researchers who could use the services that we provide. You can reach out to us very easily if you go to either website, either the Genomics Center or the MAGIC website. You can find our contact info. If you Google Genomics Center or MAGIC at Rutgers, we come right up. So please reach out to us if you have any questions. And now I'm going to turn the presentation over to Kendra and Suma from 10x Genomics.
Hi, everyone. I just want to introduce myself first as your sales executive. I've been at 10x Genomics about 3 months, and I'm the first point of contact. We have the wide range of support from Suma, who will be giving the talk to help you plan your experiments all the way through FASs at the end to make sure your experiments are going well. So I also started actually back at New Jersey Medical School, where I was in graduate school for about 4 or 5 years, and then have gone on to other labs and sales. But I'm really excited to be here at 10x. So please, I'll put my email in the chat. Feel free to reach out to me if you have any questions. But luckily today we have Suma here, who's our science and technology advisor, as I mentioned. So we're excited to have her. She's been here a little bit longer than me. But Suma started out with her PhD in biomedical engineering from Boston University. During her PhD, she was introduced to NGS and developed several genomic assays to probe the regulatory network of Mycobacterium tuberculosis. And because of her interest in technology and assay development, Suma then joined the New York Genome Center, where she witnessed the single cell revolution. So before joining 10x, she spent 2 years at BMS, where she evaluated and validated several genetic and genomic assays, including single cell gene expression assays for biomarker discovery, and then implemented them into the clinical trials. She is now the Science and Technology Advisor for the Northeast region and is very excited to support the scientific community, adopt 10x technologies, and help address a multitude of scientific questions. So without further ado, Suma.
Thank you, Kendra. Thank you for the introduction, and thank you for the opportunity to present today. So let me go ahead and share my screen. All right. Thanks once again for the opportunity, and thank you all for joining the webinar today. I'm really excited to be here to speak about our existing portfolio, as well as talk about the new product introductions in both single cell and spatial multiomics. So today I'm going to divide the talk into three parts. First, I'm going to talk about advances in our Chromium Single Cell platform, and then talk about our Visium Spatial platform, and finally give you an introduction of Xenium In Situ platform. As we all know that primarily all of us are interested in understanding how complex organisms such as ourselves with trillions of cells develop, function, and interact with our environment, and what processes go awry and lead to disease states. And this is exactly what motivates us at 10x Genomics. We want to build the tools that allow researchers like you better understand biology in all its complexity. So to address this, 10x has started its journey with our Chromium Single Cell platform to interrogate different types of tissues at single cell level. We have definitely continued to invest in this platform, and today our assays are compatible with different cell types, including different preservation methods to detect the true biological state. We have some really exciting updates today showing single cell data on FFPE, so stay tuned for the seminar for the later part of the talk. And of course, cells don't act in isolation. They are organized into tissues and act in concert with each other. So spatial information is very crucial to reveal this structure and context. Our Visium Spatial platform, it's a discovery platform that allows unbiased measurement of whole transcriptome. And this information combined with the morphological context from the same section, we are now able to bridge the gap between histology and molecular biology. Finally, we have an update on the Visium assay. Now we are able to measure both the transcriptome and the proteins. Now, while single cell and the Visium Spatial are discovery-based tools, Xenium will let us probe in a targeted and a focused manner. So we identify a set of targets using our unbiased platforms, and we can validate those targets using our in situ Xenium platform, which is going to be released at the end of this year. We can get high resolution and run as many samples as possible with this platform that gives a molecular resolution. So with that, at 10x, we innovate rapidly. We do focus not just on individual assays, but we invest in entire platforms. So when researchers are committed with our investing in our platforms, they do know that we'll be continuing development and addition of new assays and features. This is best illustrated by our Chromium Single Cell journey. It was first launched in 2016, but we've continued to innovate, release new versions of existing products, and add additional products and capabilities. And there's no doubt we'll continue to do this into the future with all our platforms. So with that, let's jump into Chromium Single Cell as a suite of assays. I'm going to provide some brief background on the core technology of how 10x works, but primarily spend most of my time on the latest new product introductions. So our Chromium Single Cell platform has been transformative for biological research. Our single cell gene expression, immune profiling, attack, and multiome assays have been adopted by scientists globally and have allowed all of you to profile more than 5 billion cells. And this is just the beginning. And recently, the Human Cell Atlas published 3 large single cell studies in Science, totaling over 1 million cells and spanning across 33 organs. This beautifully illustrates the power of these technologies to unravel the complexity of biology. So as you can see in the single cell Chromium innovation engine, from gene expression, immune profiling, and addition of feature barcoding technologies such as
protein information and CRISPR features, through to ATAC and multiome, where you can profile both chromatin accessibility and gene expression from the same cell. We have developed a suite of single cell assays that have fundamentally changed the way research is done. So today I'll present the latest additions to our single cell portfolio, including the BEAM, which is barcode enabled antigen mapping. It's our soon to be released assay for immune discovery product. And I'll talk about fixed RNA profiling. And finally, I'll talk about our nuclei isolation kit within the Chromium Single Cell space. So before jumping into these products, let me provide you a brief background on the core technology on which all of our single cell assays are based on. So 10x Genomics assays utilize a microfluidic platform to partition cells or nuclei along with barcoded gel beads into nanoliter volume droplets. And these are called as GEMs, which are gel beads in emulsions. Depending on the assay of choice, the input can be cells or nuclei for gene expression, cells stained with oligo-labeled antibodies when you're interested in measuring proteins, or cells stained with oligo-labeled antigen stained tetramers for antigen specificity experiments. Once these cells are partitioned into droplets along with the gel bead, the second step is the molecular barcoding. This step happens within the droplet, during which the barcode associated with the gem with the gel bead gets transferred onto the individual molecules of a cell or a nucleus, thereby preserving the single cell information. After barcoding, the emulsions are broken and the barcoded molecules are collected for downstream library preparation, sequencing, and analysis. Now, the key component of our single cell assay is the gel bead. Each gel bead contains millions of oligos, and the structure of the oligo varies with the assay of interest. As you can see on the left, it's an illustration of oligo structure present in gel beads for our 3' gene expression solution, and on the right is for our 5' immune profiling solution. Each oligo typically has, if you're looking on the left, each oligo typically has an Illumina TrueSeq read 1 or Illumina read, followed by a 10x barcode, which is specific to the gel bead, and then followed by a UMI, and then a capture sequence. So this capture sequence can vary depending on what molecule you're capturing. In our 3' gene expression, we are capturing poly A tail of mRNA molecules. So you see poly DT as a capture sequence. Now, you can see that there are a couple of other oligos that are grayed out in our 3' gene expression solution. And this enables feature barcoding technology. And what I mean by feature barcoding is you can capture any feature, such as protein or antigens, that cannot be sequenced directly. And the way this works is you can conjugate these features with a DNA oligo containing a sequence that's complementary to the capture sequence present on the oligos on the gel beads. Now, coming to the 5' immune profiling solution on the right, the capture sequence here in green is the template switching oligo. So the template switching oligo gets added at the end of reverse transcription step. And our immune profiling solution also allows to profile full-length VDJ sequences for TCR and BCR-seq analysis. Now, for our multiome assays, the oligo structures looks different. If you look at the bottom image, there are two types of oligos present on the gel bead for our multiome assay. If you're not already familiar with multiome assay, it lets you profile both ATAC, that's open chromatin accessibility, along with gene expression from the same single cell. So the poly DT, which is present on one of the bead, captures the mRNA molecules. And on the other oligo, you see this sequence space, which is called as spacer. It's a proprietary sequence by 10x Genomics, it's used to attach the barcoded fragments where the adapter is introduced by the transposase enzyme during the transposition. And another point to note here is that for multiome assay, the input is transposed nuclei. Transposition happens in the bulk, and then you load them up in the chip along with the barcoded gel beads. So basically, the structure of the oligo depends on the assay of interest, but the core principle is that you supply your cells, either it's cells, nuclei, transposed nuclei, cells stained with antibody, oligotagged antibody, etc. And you capture them into the gems along with the gel beads. So with that, I'm going to jump into our latest product introductions, as I mentioned, the BEAM product, Nuclei Isolation Kit, and Fixed RNA Profiling Kit. The first one is the BEAM product. So BEAM is barcode-enabled antigen mapping. What it lets you do is it enables simultaneous sequencing and identification of antigen-specific T cell and B cells. And this is built as an extension of our immune profiling product that I just described a couple of slides ago. And as I mentioned, our immune profiling product allows us to sequence full-length TCR and BCR sequences. And another thing to note is that current processes for immune cell discovery are really slow, low throughput, and inaccurate. So this is where BEAM really comes into picture and will add value, as it lets you screen theoretically unlimited number of antigens in a single experiment and identify T cells and B cells specific for a given antigen. So why is it so much faster? Because it's directly built on our immune profiling workflow. If you have already have experience with that, it's a very robust, streamlined workflow. And on top of that, all you need is a 5' antigen barcode kit for BEAM antibody, or a 5' MHC class 1 kit for BEAM-T to enable capture of antigen-specific B cells or T cells. And what this means is scientists can potentially screen thousands of antigens in parallel in a matter of days from one particular sample. And unlike other technologies, you can obtain a high-resolution cellular portrait that includes information on gene expression, and if you want surface marker identification along with TCR, BCR sequence data. So what does that entail? What do you need? What kind of supplies are required? So what the 10x will supply the antigen barcode kit that basically contains a uniquely barcoded streptavidin PE complex. So for BEAM antibody, the customer only needs to provide cells and biotin-related antigens. And the 10x will provide, as I just mentioned, the uniquely barcoded streptavidin complex. So you mix that complex along with your biotin-related antigen, and you combine with the cells for downstream analysis. For BEAM-T, we are leveraging our acquisition of Tetramer Shop. So the MHC class 1 kit will contain monomers for you to load your peptide of choice. And it also has, similar to what the BEAM antibody kit has, the BEAM-T also has a streptavidin labeled complex. And the loading of empty MHC monomers is very rapid, easy, and reliable. So once you're able to load these streptavidin complexes with your own biotin-related antigen for BEAM antibody or unique peptide for BEAM-T products, all you have to do is combine them with the cells and sort through, do a flow sorting to enrich for antigen-specific B cells or T cell population. Once you enrich for those populations, you encapsulate the cell population into gems using our Chromium immune profiling workflow. The results or the data will be viewed and analyzed on our soon-to-be released software. So as a data highlight in this data set, PBMCs were isolated from COVID convalescent patient contain, and it's expected to contain different immune cell type diversity. So BMT reagents are then assembled with the peptides of interest for COVID, as well as flu, EBV, and CMV viruses. As you can see on the plot to the right, that BMT labels CD8 effector memory, as these are T cells that have developed specificity for all of these antigens, while the naive T cells, as the name implies, have not acquired high specificity for any of these antigens. So it basically reflects the power of this product for simultaneously screening multiple antigens and to be able to get the entire phenotypic information of antigen-specific B cell or T cells. So to summarize, our Beam antibody and BMT product will be available in late Q4. It will enable discovery of antigen-specific B and T cells in a matter of days from one sample. And you're able to obtain high resolution cellular portrait that includes surface marker identification, gene expression, and BCR TCR sequences. As with any other products, this kit will also come with analysis and visualization software, and it will complete the end-to-end workflow, and it will support the visualization of Beam data on a per antigen basis and provide Beam-specific sequence information. Moving on to the nuclei isolation kit. Just as we want to expand the functionality of our core single-cell technologies, we also aim to unlock as many samples for as many customers as we can. And we have heard from all of our customers about the need for having a robust nuclei isolation protocol so as to input them into our attack and multiome solutions. And we also know that there's large number of frozen samples that are archived, and because the existing nuclei isolation protocols are too low throughput or variable and are not really robust, it's been a pain point for many of our customers. So here we introduce Nuclei Isolation Kit, which is a simple, scalable product for nuclei isolation from fresh and frozen tissues. We made the workflow as straightforward, as robust as possible. The tissue is dissociated in an optimized lysis buffer, and then it's passed through a nuclei isolation column. The isolated nuclei are then resuspended in a debris removal buffer, and washes are performed to remove debris. Then once you have the final nuclei suspension, the nuclei are counted and you can input them into any of our 10x Genomics single cell solutions, including gene expression, attack, multiome, or even the fixed RNA profiling that I'm going to talk about in a bit. The streamlined workflow allows for processing up to 8 samples in parallel and in just about an hour. So all you need is an hour of your time, a benchtop centrifuge, and an interesting frozen sample to work with to use this product. We compared our new nuclei isolation kit performance to other widely adopted methods in the field, and we see a really amazing performance of this kit, which is better than the existing methods. So this is just a data highlight that we've used our nuclei isolation kit on human kidney tumor. Having a kitted solution for nuclei isolation means that you don't have to spend time on optimizing this isolation protocol or don't have to waste precious samples. So here we show the multiome data set, and basically shows that the nuclei isolation kit is able to preserve all cell types in both attack and gene expression assays. So we have vetted this protocol across almost 30 various human and mouse tissues, and we have seen similar outstanding data quality without having to optimize a bit. So all of these tissues are processed with our kit out of the box, requiring no optimization. And this demonstrates the breadth of applications that are enabled with this nuclei isolation kit. So basically, with our nuclei isolation kit, we have built a complete product for nuclei extraction that's easy to use and allows us to generate high quality nuclei suspensions without any expensive instruments or complicated pipetting. As I mentioned, the workflow is applicable to a wide range of tissues, and it allows capture of even sensitive cell types like immune cells, and allows you to process up to 8 samples in parallel in just about an hour. And more specifically, this is optimized specifically for 10x Genomics assays, and we are really excited to see how customers are going to adopt this in their workflow and generate valuable information from our multiple assays in our portfolio. Switching gears to single cell fixed RNA profiling now, we have multiple single cell assays in our portfolio, and we are just getting started in the sample preparation space. Even with the latest nuclei isolation kit and multiple demonstrated protocols on sample preparation, there is still a gap in sample handling logistics. So in order to make the sample handling logistics easier and to stabilize samples so they can be collected and batched for larger studies, and to improve single cell data quality on the most difficult and least stable samples, we are introducing single cell fixed RNA profiling solution. Like any other assays, this is a fully validated, kitted, robust workflow that includes everything from a demonstrated protocol for sample preparation, fixation and storage through the final data analysis. One point I want to note is our fixed RNA profiling solution is only able to run on our latest Chromium X series of instruments, that is Chromium iX or X. The chip is completely redesigned, and now the fixed RNA profiling workflow supports ChipQ. And this assay can be run in either singleplex format, where you can recover up to 10,000 cells per channel, or the multiplex format, which is really a highlight of this product. So you can multiplex up to 16 samples per lane and recover up to 128,000 cells in just a single channel. The rest of the features, such as microfluidic cell capture rates or the multiplet rate, remains the same as compared to any other single cell gene expression assays. And this is also compatible with feature barcoding technology, but for now, this is only compatible with, for cell surface protein in the singleplex format. So as I mentioned, this is a completely validated kit. And now let's look at the workflow in the singleplex format. So in the singleplex workflow, the starting point is dissociated cells. I'm going to speak a little bit more about what are the starting sample input for this workflow. But in the singleplex format, you have an option to stain the dissociated cells with antibodies to probe cell surface protein expression. And this is compatible with our total seq-B antibodies supplied by BioLegend. So once you stain the cells, if that is of interest to you, you then fix the cells with 4% paraformaldehyde and permeabilize them. After that, you have an option to store them. Once you've fixed and permeabilized the cells, you then use gene-specific probes and are hybridized to mRNA molecules. So unlike our existing gene expression workflows, we are not capturing the poly A tail of mRNA, but instead we are using gene-specific probes that get hybridized to the targets. So once a hybridization is performed overnight, the unbound probes are washed out and cells are partitioned into gems along with the gel beads. Now, inside each droplet, the hybridized probes are ligated, barcoded, and then downstream PCR completes the sequencing library. In the multiplex workflow, the steps are largely the same, with the exception that up to 16 different hybridization reactions can be performed. And each hybridization reaction is performed with a uniquely barcoded pool. Once a hybridization is completed, you can pool all the samples, wash and remove unhybridized probes, and partition, and the next steps are exactly the same manner as I just described in the singleplex assay. In both assays, the probe design is very crucial and paramount to the success of the assay. So as you can see, for each mRNA target site, there are 2 probe pairs designed, the LHS probe, which is left-hand side probe, and RHS, which is right-hand side probe. So the left-hand side probe contains flanking sequences to accommodate Illumina sequencing workflow. And the right-hand sequence consists of a constant sequence along with a probe barcode, which is what differentiates or adds barcode to each of the mRNA molecule, and the gel bead capture sequence on the flanking sequence of the right-hand side probe. This is exactly what enables multiplexing of so many cells. For those of you who may have experience with cell hashing or cell plexing workflow, in those workflows, you sequence multiplets and you get rid of the multiplets because you cannot differentiate which mRNA has come from which cell. But here, each of your mRNA molecule is barcoded. And what that means is you don't have to throw away the multiplets anymore, until those molecules are not coming from the same probe barcode. So that's the beauty of the fixed RNA profiling workflow and how it enables multiplexing of up to 16 samples and to get 128,000 cells in one channel. So the probe set is designed for the entire transcriptome for approximately 18,000 coding genes in human and 20,000 coding genes in mouse. And each gene has 3 sets of pairs. So this allows you to increase sensitivity and robustness of the assay. The fixed RNA profiling workflow enables multiple stopping points. The first stopping point is once you fix the dissociated cells, you can store them at 4 degrees or -20 or -80 degrees, depending on how long you want to store them. Excuse me. I think so far we have validated up to 3 months, and we are continuing to validate the storage time, and we'll update you as we get latest data. And the second step at which you can stop the workflow is after hybridization. So post hybridization, you can store the samples at -20 and -80. You can store them as pooled or individual samples, and that allows you to mitigate experimental risk. And you can go back to that hybridization cells if you want to continue to doing the analysis. And if you want, you can analyze more cells after initial analysis. So currently, our assay is compatible with human. Mouse is going to be released soon. And going back to the input sample compatibility question, the assay is compatible with cell lines, primary cell suspensions, isolated nuclei, dissociated tissue samples. But we are also adding the capability, which just got announced last week at AGBT. Now you can fix the whole tissue as it is and then store it and then take it into single cell or single nuclei dissociation and take it into the fixed RNA profiling workflow. Just as a data highlight, here you can see the assay comparison between the fixed RNA profiling and our 3' version 3.1 assay. Because there are 3 probe sets designed for each gene, the sensitivity is way higher. It's higher than our current 3' gene expression assay. And on the bottom plot, you can see the median number of genes per cell is higher, if not the same, as compared to our 3' version 3.1. Currently, we have different kit sample plexing options available. You can choose to do 1-plex, 4-plex, or 16-plex, and the way we are having our kits available. And on the bottom plots show that no matter how many pools you multiplex, you're not losing sensitivity in terms of your UMI counts or the median genes per cell detected. And this is another data highlight showing exactly the same thing. Multiplexing distinct biological samples, patient cohorts, time points, or different biological tissues are all important use case for sample multiplexing using fixed RNA profiling. However, as with the colon tissue example shown earlier, multiplexing can also be used to increase the number of cells on a single sample while controlling the multiplet rate. So in this example, it's another example for multiplexing. We have multiplex 16 subpools of a healthy human PBMC sample and targeted around 5,000 cells per pool. So we recovered 80,000 cells in total and with an approximately 4% microfluidic multiplet rate for the entire 5,000 cells detected per sample. This is really exciting update that just actually got announced last week at HEBT. FFPE sample type is now compatible with our fixed RNA profiling solution. The protocol is very preliminary, but please let us know if you're interested in testing this protocol. And I don't have to say the importance of FFPE preservation, and it's a standard method of preserving across many samples, especially in like clinical trial samples and surgical pathology specimens. Since these are the same samples used by pathologists, the metadata for these patient samples are also quite rich, opening the door to much more comprehensive analysis that are possible even with the smaller cohorts. And that's why we're really excited today to share some of the preliminary data on FFPE sample type compatibility. So this, here we used a block of invasive carcinoma. We used two sections of 25 micron thickness, and using a combination of manual dissociation and enzymatic digestion, we took this into our fixed RNA profiling solution. So when we clustered the data, it was really exciting to see that we were able to preserve all cell types, including different immune cell population. So, so today I've talked to you about 3 products in the Chromium Single Cell space, and really excited to see what customers are going to do with all of these products. Now, switching gears to Visium Spatial analysis, as with our single cell Chromium platform, we are constantly advancing our Visium Spatial assay. Our ultimate goal is to develop tools to enable you all to make new, previously inaccessible biological discoveries. And I love this animation of mouse embryo where you can see how each of the single cell back mapping onto the embryo section. This is, I think, the power of Visium Spatial. Our Visium platform began with just a single cell product in 2019, where it was developed for fresh frozen. And since then, we have expanded and added capabilities of immunofluorescence just to measure a handful of proteins. And then we added Visium for FFPE. Today, I'll present a few recent developments on our FFPE platform, along with Visium Site Assist. It's a new instrument for bridging the histology and molecular biology workflows. Our Site Assist instrument makes running the Visium assay even simpler by allowing you to section the FFPE block on standard glass slides. And I'm going to talk more about this in just a minute. Our Visium platform leads the way in spatial biology. This is exemplified by the number of institutes that are using our Visium products. And the productivity of the researchers at these institutions as measured by rapidly increasing number of publications and preprints. Before I jump into the latest updates, I want to make sure we all are on the same page on how the spatial assay works. So while molecular barcoding happens within droplets for our single cell solution, it happens on a Visium Spatial slide in our spatial assays. So in the Visium workflow, fresh frozen or FFPE tissue section is placed on our Visium Spatial gene expression slide as shown on the left. Each slide contains 4 capture areas, and each capture area contains approximately 5,000 barcoded spots. And the capture area size is around approximately 6.5 by 6.5 millimeter square and is surrounded by a fiducial frame that you see in red. That's used for image alignment. And each spot diameter is 55 micron and the center to center distance is around 100 microns. So what this means is it lets you profile from anywhere from 1 to 10 cells per spot. If you zoom in to each of these spots, it consists of high density lawn of spatially barcoded probes. The structure of this probe is very similar to what you have seen for our single cell solutions. It contains an Illumina partial read 1, spatial barcode unique to each spot, and a UMI unique to each probe. And finally, the poly-DT region to capture poly-A of mRNA molecules. I do want to make sure that the workflow for FFPE is slightly different. For fresh frozen, you're going to permeabilize the tissue sample and the mRNA molecules are released and the poly-A is captured using the poly-DT tail on the oligo. However, for our FFPE workflow, we are using probes similar to what you have seen in our single cell fixed RNA profiling workflow. So here, probe pairs are designed across the protein coding transcriptome, 18,000 genes in human again. And the probes are hybridized to the mRNA target sequence and are ligated to one another. Then the target RNA is digested and the target and the ligated probes are released. Now, if you look at these right-hand side probe, it has a poly-A tail. So once these probes are ligated and released, the poly-A tail is used to capture these molecules onto the Visium spatial slide. So we are basically sequencing the probes and not the mRNA molecules. So with that, let me talk more about our instrument Visium Site Assist, along with providing updates on 2 other assays. There's some assay improvements for our manual Visium for FFPE version 2. And then we are finally introducing our spatial proteogenomics assay that lets you profile both gene and protein expression. So before going into Visium Site Assist, in case you're not familiar with the FFPE workflow, there are few challenges in the FFPE workflow in the sample prep part of the workflow. So FFPE section has to be freshly cut and must be placed directly on the Visium slide in this workflow. So what this means is we are unable to access already sectioned slides. And secondly, we are asking pathologists to learn new set of skills such as placement of tissue directly on the Visium slide. And this can be a little bit tricky depending on your skill set. And you basically only get one chance to place the section correctly. So to overcome these challenges, especially that histology labs might not be equipped to adapt their tissue preparation methods, and they might not be comfortable working with NGS-based assays. Conversely, the genomics labs have limited experience working with tissues. So to bridge this gap, we are launching Site Assist. So what does it do? With the Site Assist platform, you can now leverage existing histology workflows. The pathologists don't have to get trained on placing the section on a Visium slide. Now, pathologists can continue to do what they're really good at and what they are specialized in. You can access tissues that are pre-sectioned on the standard glass slides. So you can just take the standard glass slide and input it into our Visium Site Assist workflow. Simplified workflows allow for easier collaboration with both the cores, and it will enable multi-site projects. And CytAssist enables workflows and allows for flexibility with various tissue sizes, which I'll come to it in a bit. So in the CytAssist workflow, the input is basically 2 tissue sections, which are on standard glass slides, and 1 new specially designed Visium slide. And this Visium slide has 2 reaction chambers. Once the slides are loaded into the instrument, the CytAssist controls the process of tissue permeabilization. So analytes are released from the tissue and then transferred onto the Visium slide on the bottom, as seen in the image. CytAssist ensures that the spatially resolved gene expression patterns can be mapped back onto the tissue section by capturing an image of the tissue relative to the fiducial frame of the Visium slide. And in our analysis pipelines, the images taken from CytAssist, as well as the images taken upfront with our H&E or immunofluorescence images, these are both combined and utilized in our downstream analysis for spatial orientation of the image. This is just a data highlight using CytAssist. So during the development of CytAssist platform, we have made sure and we have spent a lot of effort to comparing gene expression clusters detected in the FFPE tissues section directly on the Visium slide compared to section on the standard glass slide. So on the top panel, you see the H&E image on the left, and on the right is the gene expression clusters detected when the tissue section is placed directly on the Visium slide. On the bottom panel, you see H&E image on the left and tissue section placed on a regular glass slide and processed with CytAssist. And it's really exciting to see that CytAssist captures spatial expression patterns without compromising any sensitivity and allowing seamless integration of standard section blocks into the Visium assay. And this is another data highlight using a 5-month-old FFPE section from a human lymph node. On the left, you see H&E image. On the middle, it's a whole transcriptome gene expression using our Visium assay. And on the right, you can nicely see that see the spatial pattern of our RGS13. It's a marker for B cells in germinal centers. Again, highlighting the sensitivity performance using our CytAssist assay. So using the CytAssist, tissue preparation for Visium has become much more flexible. Whether it's in the clinical trial sites, sending recently mounted tissue sections to a central site for processing, or using sections that have been stored on a slide, or collaborating with the histology core services to prepare sections from existing blocks. All of these combinations have now become possible with Visium CytAssist. The instrument is expected to be available approximately next month, and this will help expand sample access and simplify sample management for our Visium assay for FFPE samples. So Visium for FFPE, launched last year, has proven to break spatial barriers in a number of research areas, including oncology, immunology, and neuroscience. However, we have hearing from our customers some of the challenges that are included with our FFPE assay. So we have made several advancements in our version 2 of the kit, where there are changes made to the slide so that the placement of tissue section is now more robust than our version 1 of the assay. Secondly, we have made changes to our probe set. Now it's similar to the probe set that's used for our fixed RNA profiling solution. So which means instead of 1 probe set per gene, we now have 3 probe sets per gene. So that increases the robustness and the sensitivity of the assay. So what this means is earlier our cutoff for tissue blocks to input into the assay is DV200 of greater than 50%. For those of you who don't know what DV200 means, it is it's percentage of fragments that are greater than 200 base pairs. Now this QC has gone down from 50% to 30%. And finally, we now are coming up with slides that have larger tissue area, larger capture area of 11 by 11 millimeter square compared to 6.5 by 6.5 millimeter square. This will let you probe larger tissue sections in the same experiment. So it's just an example where we are able to profile a larger tissue section from FFPE human breast cancer tissue section mounted correctly on the Visium slide. Lastly, we are introducing our spatial proteogenomics assay where you can profile both protein expression and gene expression from the same FFPE section. There are competitive technologies where they can obtain this information, but in a serial section. In this assay, we can get protein, RNA, and morphology from the same FFPE tissue section. This assay is also compatible with H&E and IF staining. This protein expression is a sequencing-based readout, and it's a highly multiplexed quantitative protein detection. We now have a panel for human immune cell profiling. It contains approximately of 35 antibodies. And if this is something of interest, please reach out to us and we can provide more details on the panel. This is just basically showing that the Visium for V2 slide has oligos for capturing both RNA and the capture sequence for protein. This basically demonstrates the sensitivity of when the protein is added. When the protein is added, you're not losing any sensitivity performance on the gene expression. As you can see plot on the left, the median number of UMIs per spot remains the same for both gene expression assay as well as gene expression combined with protein readout. And on the right, you can see consecutive sections from the same block using our pan-CK protein marker that's included in the panel. So lastly, just want to reiterate that you can get three informations from the same section: morphology on the left, protein in the middle, which is more robust compared to the gene expression on the right. And both the right panel RNA shows unsupervised protein and gene expression clustering obtained from the same section. And we can also look at immune cell markers such as CD3E for T cells, CD19 for B cells, and CD163 for macrophages. On the left is the protein data, and on the right is the RNA data. And it's not surprising to see the robustness at the protein level compared to the RNA level. In the interest of time, I'm just going to go into the Xenium In Situ. I'll just provide a brief overview of Xenium In Situ. And as I mentioned earlier, please reach out to us for more information if this is something of interest. And while the single cell and Visium allows us to do unbiased whole transcriptome, and these are discovery-based tools, Xenium is a tool that is a focused and allows us to do target validation. So using our In Situ Xenium platform, we can do targeted and focused measurements of genes that are of interest, and we can obtain subcellular molecular resolution with high sensitivity and high throughput. And we are just at the beginning of our Xenium journey. And as you have seen with our Chromium Single Cell and Visium Spatial, we have no doubt that we are going to have a very solid roadmap for our Xenium assay, and we'll develop continuous updates and add more features to our Xenium assay. So what does our platform look like in its current format, which is soon to be released at the end of this year? And how does our roadmap look like? Currently, we are planning to release curated panels for specific tissues. There is a little bit flexibility to add custom content. The assay, when it's going to be released, will allow you to measure RNA and can let you measure up to 400 genes and up to 6 slides of 12 by 24 millimeters each per week. And you'll have an onboard analysis software, nuclei-based segmentation, and a Xenium Explorer to explore the data yourselves. In the roadmap, we are going to add more menu of tissue and application panels. And we'll be able to do simultaneously profile both RNA and protein from same slide. And we are looking to include SNP information, isoforms, and etc. Excuse me. And there is a flexible plexi to suit your application needs. And we will be looking to increase higher throughput per week. And more features will be added in our software analysis, including cell segmentation, simplified storage, in collaboration with 10x Cloud. So the initial commercial release of Xenium platform, as I mentioned, is expected later this year. And we're already excited to see what you'll be able to achieve with this technology. This is just the beginning of our roadmap. And as I mentioned, like our Chromium and Visium platforms, will continue to build this foundation and enhance our core capabilities. Finally, I would want to leave you with the data analysis slide. As with any other product, we have a complete suite of tools, including Cell Ranger for Chromium analysis, Space Ranger for Visium, and we also have Loop Browser for visualization analysis, and we also have cloud analysis that is available for single cell currently. And it's not surprising that the number of publications are exponentially increasing. And we are very excited and motivated to make new and improved products to help you push science forward. With that, I would like to thank all of you and leave it on this slide, the biology's most comprehensive toolkit. Thank you. Thank you so much for joining today.
Thank you for a great talk, Suma. We do have a couple questions already in the chat. So if anybody else has any additional questions, please feel free to put them in the chat and we will answer them. But we have two that look like they're about the FFPE spatial platform.
Mm-hmm.
The first is the ATAC and gene expression kind of combined available for the FFPE spatial?
Not at this moment, no. But there are some advances for ATAC and spatial. This field is exploding outside of 10x. So if that's something of interest, I can definitely work with you to see what's the right product, right technology that's available right now in the academic space to use into your research. Okay.
And we have another question for the FFPE spatial. Can you process FFPE sections that are 10 plus years old on that platform?
That's a great question. We all know that once the sections are placed, the quality of RNA will degrade with time, but we can definitely extract some of the RNA and see what's the RNA quality. If as our QC indicates, if the DV200 is greater than 30%, we can definitely give it a try. I think there'll be more recommendations on the age of sections that have been tested. I can definitely get back on what is being tested, and you can definitely try it. Yeah.
Okay, cool, thanks. I guess we'll see if we got another question come in. Is single cell proteomics based on antibody detection or mass spec?
Is single cell proteome, it's antibody detection, but the antibody is conjugated to oligo. So it's a sequencing readout.
Okay, perfect. And I guess, if anybody else has additional questions, please feel free to keep sending them. I have a couple as well. So one kind of on the Visium as well. So before, the spots have kind of previously been a little bit on the larger side where they can capture multiple cells per spot. Have they gotten to the single cell resolution yet, or is that kind of in the roadmap soon?
Yeah, it's definitely in the roadmap. It's expected to be released at the end of the year. I think we're facing a few hiccups, but that are being addressed, and it's definitely on the roadmap.
Awesome. That'll be awesome to see. And I guess while everyone else is thinking of questions, I got a couple more. So for Beam, which is something I haven't seen yet, and I think that's really cool because it's kind of like the evolution of the Immudex barcoding that was kind of generated previously. I know you mentioned that you could do thousands of antigens at a time. Has there been a tested limit yet? And like, what is the barcode length that you're using for those?
Yeah, so this is, we don't have a lot of details on this yet. I'm assuming it will probably, the barcode length is probably going to be similar to our other feature barcoding technologies, but we can definitely provide more details as we as we generate more data and release the platform commercially. So your previous question was how many antigens tested out, right? Yeah, it's still evolving. We have no information on that yet.
Okay, cool. That'll be cool to see kind of what the upper limit is. 'Cause I know for feature barcoding for a while, they were like, yeah, we don't have an upper limit yet. We'll just kind of see what happens.
At least in the protein space for the site-seq, I have seen a max of maybe 284 antibodies. But yeah, I'm excited to see how researchers are going to use Beam product and what kind of antigens they're going to screen.
Yeah, it's always exciting to see what's new.
Yeah.
Here's another question. When will the fixed RNA profiling platform be available for mice?
It's soon. It's probably going to be in the next quarter, but we don't have the exact date yet, if I'm not mistaken.
And are there other, like, I mean, I guess common ones like rat and zebrafish, are those kind of in the roadmap down the road as well?
Yeah, so we are collecting feedback from customers on how they're going to use our fixed RNA profiling and what's the top request. So based on that, we will direct those requests to our product manager team, and then they can make a decision accordingly. So keep the requests coming if you have interest in other species, and Kendra or I can channel that request back to our product management team.
Okay. Perfect. Okay. Well, I guess if anybody has any additional questions, feel free to reach out to myself, Pat at the Genomics Center, or Kendra and Suma from the 10x team. I'm sure any of us will be happy to answer the questions or put you in touch with the correct people, as needed. Yeah. Again, just thank you for the 10x team for presenting this excellent presentation today, and thank you all for attending.